All right, I guess we can get started. Welcome, everyone. I'm very excited that you're all here for our gene delivery seminar series for July. We are very excited to have Dr. Alan Griffith with us today. Alan is the head of CDMO Global Operations at VectorBuilder, and he has been working in the CGT space for over a decade. Alan was the fifth employee at Mira G, where he went on to help design and build London's first subterranean end-to-end multi-vector, multi-product GMP facility. He then worked at the first kidney-centric GT company, Pure Spring, as the vice president of research and led the team to selecting a novel AAV serotype capsid and generated a novel kidney-specific promoter as part of the clinical candidate selection process. He has been instrumental in enabling 5 to 6 clinical trials across 3 startups in 5 different disease areas. In his career, he has held many senior positions, such as Associate Director, Vice President, and Chief Development Officer, and has come full circle to work in the CDMO space. Alan is keen to support VectorBuilder's clients in avoiding some of the overlooked pitfalls in CMC, QC, and process development. He is an advisor for many industry groups and consultants for developers and focus groups. In this webinar, Alan will set the scene regarding the global CGT landscape and summarize the pain points of CGT manufacturing from the perspective of a contracted developer and manufacturer. With that, I am going to hand it over to Alan. I think he is going to talk you through the wonderful things about CliniVec. I want to mention that this webinar is being recorded and will be available later. Alan, you can take it from here.
Thanks, Amita. Thanks so much for the kind intro. Well, good morning, good afternoon, good evening, everyone. It's a privilege to be here to talk about, CliniVec on behalf of VectorBuilder. As a whole, we're at a very critical inflection point, I think, in the field right now of cell and gene therapy. The moment where we're really seeing this boost to coming into clinical trials, is there enough tools available to everybody in order to help them transition on that journey? That's part of the conversation we're going to have today around CliniVec. A bit of history, I suppose, since the early 2000s. We've seen incredible breakthroughs in cell and gene therapy, but we've also been faced with sobering lessons, things that we could have done better. Of course, go back as far as 1999 with the death of Jesse Gelsinger, highlighted basically how important taking your time in vector design, taking your time in manufacturing is so critical to the safety of getting these wonderful transformative therapies across the line. That's the beauty of them. They work, they're curative, they're not a long-term treatment, they're a one and done type modality. Basically understanding the complexity and risks around vector design and how that impacts the safety profile, the manufacturing profile is essential to people's success going forward. And we're proud to be the only CDMO capable of rolling out a CliniVec type service, and that's what we're going to try and talk about today. Moving on. Before I go any further, a quick lesson. Again, this is more so to make it available to people offline. The session is recorded, so it will be good, obviously, then to look back at the history. I won't talk you through every aspect here, but the main message being, we're at a culmination now of 60 years more of research. There's been an early days, or the Stone Age times of cell and gene therapy, where early patients were getting these treatments, and on the onset of Watson and Crick, obviously coming up with the DNA helix, we've been able to decipher the genetic code in such a way that we can deliver gene replacement therapies where genes are being defective or genes are null in certain pathologies. We can actually use viral vector machines or vehicles in order to put those genes back in. So we've come a long way. What's happened the most, I suppose, over the last 20 years, where 15 of these 20 years I've been involved in, we've seen exponential growth. And again, that's a testament to the kind of investor community getting involved in it, the success, how the science has evolved, and how more and more people are getting involved in cell and gene therapy. It's the fastest growing subsector of biotech. So, and it isn't slowing down anytime soon. And we're seeing now, even the next 2 or 3 years, there's 10, 20 products going to come through. So that exponential growth is simply continuing. So it's an exciting time to be involved. And I think we're now coming up with CliniVec pretty much at the right time, and it's really why, that's why I have the vector roulette design symbology, because it's about not really leaving your vectors to choice. Or to chance. It's about stacking the game in your favor or hedging your bets. And I think, that's really, really important to try and take home message. This is the history continued again for our offline kind of visibility. I think one of the main things is to see the short space of time between all of these different vectors getting to commercial acceptance or BLA. They're being accepted then for commercial. To give you an idea, cell and gene therapy, they have about 28 commercial products, where small molecules have thousands. So you can see we're still in the immature stage of it as a biotech sector. But ultimately, we're hoping now CliniVec can actually help accelerate a lot of this. Okay, and then before we get deep into it, there is two or three concepts I really want to try and explain, because they're acronyms. And the same thing with acronyms. Sometimes, obviously, for people that are in the space a long time will know them, but just on the basis that not everyone will start off on the same level footing. There's three concepts I'd like to discuss really, and that's first one being CMC. It's basically regulatory acronym for manufacturing, a fancy word for manufacturing. It stands for chemistry, manufacturing, and controls, which I'll be saying the word CMC pretty frequently. I'll be also using the word IND pretty frequently. This stands for investigational new drug. And basically what that means is you normally go through the discovery stage, preclinical stage, get to clinical trials, you generate enough material, and you have an IND submission, then ultimately going into phase 1, phase 2 clinical trials. I've used a very simple schema here just to try and explain that, you cannot go into clinical trials without an IND submission. And in order to generate that, there's a regulatory requirement that you have certain prerequisites that you need to hit for that. CMC, that's why the arrow is pointing at CMC. That's simply just one aspect of the IND submission. It's called a module 3. It's basically encapsulates all the manufacturing and quality control attributes for your drug product. And then the final concept, vector. VectorBuilder by name, VectorBuilder by nature. But we obviously, during CliniVec, I think consultation, we realized that CliniVec is not just the vector, it's actually what's inside the vector, the payload, the configuration that goes inside it, which gives the therapy its potency, which gives us a therapeutic efficacy. So basically, they're the three concepts. And again, it's good just to remember those throughout the presentation. The best way to explain, I think, where CliniVec fits in is to actually talk you through the road of ATMP development. Again, ATMP being an acronym we use frequently. Again, what we'll try and do now is talk you through the cell where it goes through discovery phase into preclinical. What happens at preclinical? That's where you generate your proof of concept. It's where you generate some early preliminary safety data. Hopefully find out your mechanism of action, your MOA. It's worth mentioning that some of these cell and gene therapy products are so unique or rare that the mechanism of action is not actually fully understood in some cases. So the data still works. It's also very plausible that you can get all the way to phase 3 clinical trials without fully understanding your mechanism of action. But as long as your product is safe, the regulatory agencies really do not mind. As long as it's safe at a certain dose, your MOA can still be kind of figured out on an ongoing basis. It doesn't need to be figured out immediately. But obviously the earlier the better for your own sanity and to manage your own business. And then of course animal work. There is the Modernization Act now kicking in, which I think CliniVec can also help fight against, where the Modernization Act is all about reducing, replacing and refining the animal testing. It's about reducing animal testing going into these clinical trials, which obviously is warranted. Whereas if you have better design around your vectors early stage, you will probably have to do less animal testing. So I think the fact that CliniVec actually can help with a lot of these different attributes is fundamental at an early stage of your development. This is the number one inflection point. You want to get to candidate selection. What does that mean? It means vector selection. What vector are you going to choose going into after your animals, obviously, into your animals, into your humans. So that's essentially a go, no-go decision. Many developers or researchers would fail maybe at this stage, or get to this point and then have to go back and repeat or resurrect the program. So it's a real kind of inflection point that can make or break many different companies. Um, Obviously, then you're going in, you have that data and you've got your candidate selected, you'd go towards your IND. What you do there then is you have to talk to somebody like us. Again, you could be one of the rare companies or developers that have manufacturing available in-house. There's two modes you could work in. You could actually outsource the manufacturing, so work in a virtual way, or you could build your own manufacturing facility. Luckily enough, during my career, I've been part of building our own in-house manufacturing capability to GMP, which is good manufacturing practice. We built that in central London, and basically I've worked in that mode, and I've also worked in the mode where you've had to go to a third party, a large CDMO, a Contract Development Manufacturing Organization for the acronym. And basically that means where you need to do your manufacturing, the CMC bolted onto your proof of concept, bolted onto your safety studies, bolted onto your animal data. The next stage, you have to know, can I manufacture this? The main message here is if you can't, you might have the best therapy in the world, but if you cannot manufacture it, well, then you don't have the therapy because you can't scale it up in such a way that you can achieve a certain amount of, say, vector particles for a certain dose. So that can be another go, no go decision. So that happens. The earlier you engage your CDMO, the better. And so people come to me sometimes. It's too early to have this conversation. Honestly, that's incorrect. It's never too early to have that conversation because one, we can introduce consultation and advice around CliniVec, or two, make sure that you do these three discovery steps or preclinical steps a bit more appropriately. So then that way you're not caught out at a later stage. Going into clinical trials, then, of course, that's what everyone tries to achieve, as any good researcher or anyone working on pathologies or treating diseases will try and, that's their holy grail, getting to clinical trials. Obviously, the schema is quite simple, but it's very difficult to get there. You have to look at safety, pharmacokinetics, efficacy around dosing, dose escalation. Again, you might have a product that works really well, but if you get the dose range wrong, you might not get any efficacy and you might get no data. So dose dependency is so critical to the success of these as well. So that's really important. And then obviously being successful, you're hopefully get all the way to commercial. Again, this has only happened 28 times in the last 60 years. So yeah, but again, a lot of companies spending a lot of money, spending a lot of time and effort, and putting their blood, sweat, and tears into making these products really transformative. And then, yeah, just the final bit, CMC selection. It's all about that CMC that I told you about. The manufacturing process development should begin early. However, it's often not considered. This is the trend I've seen over the last 15, 20 years. It's an afterthought. Well, of course, anybody, once we hand it over to them, they can manufacture it. But sadly, manufacturing concerns are one of the two biggest bottlenecks in products failing or going into the valley of death. So I'll talk a little bit more about that in the following slides. Yeah, so the pipeline, again, the pipelines are growing, but the success rate is not. So we're getting more and more products coming out of academic labs, institutions, startup companies coming out of stealth. But unfortunately, the success rate is pretty much the same as it's always been. Your huge 90% of clinical trials are failing due to predominantly two things, poor CMC and poor clinical performance. Again, I've put the exact schematics just here, but as you can see, the main 90% is something that we can control. There's 10%, I suppose, that we can't. Like the lack of commercial needs, that's up to obviously the reimbursement strategies, the economics around cell and gene, that's for them. We're the scientists, we're the manufacturers. We can only kind of control 90% of the controllables here. And yeah, look, as the schema, without going through it, you can see that preclinical, there's loads of initiation of projects. There's plenty of projects going into phase one and roughly around 300 every year. Phase two, again, more or less the same attrition rate, but then we really see this drop off then going into phase three. And this has been really attributed now. FDA are really keen to get complete response letters out, which means we can really transparently see that there's main issues, and these same issues keep popping their head up time and time again. And that would be poor manufacturing, CMC, and poor performance. Just another way to look at the same kind of thing visibly. You can see here that as you're going to, and I always use this swimming lane comparison because it always comes back to that swimming lane. Vector design using non-compliant components or unregulated sequences. This is what tends to happen. A lot of startups or a lot of academic institutes historically used to have a product that worked really well in animals. And then they got handed over to a CDMO. They realized then that it's not compliant for clinical purposes. It's not suitable. It has unanswered or unregulated sequences in it. And again, there's a saying that we say, don't accept gifts from strangers, because often you can't control the provenance or the origin of that material. Again, it seems simple. It's simply someone handing a material or a vector to someone else and saying, this is now yours. Please take it to the next level of your company. But if you don't do the due diligence around that, that can lead to poor vector design, leading to poor vector safety and efficacy and compliance issues later on. Um, Again, that's just focusing on vector design, but the second one, CMC and manufacturing, you can really have, if you rush through preclinical, you'll have a severe lack of understanding around your CMC. That means you don't know how your vector will propagate, how it will amplify, and amplify at scale. You might have carryover of unwanted impurities. You might have a poor empty versus full ratio in the case of AAV. You might have integration events happening due to, if it's lentivirus. So really agnostic to the vector type. It doesn't really matter. These two issues manifest regardless of the modality, regardless of the viral vector that's used. So that's really important to kind of remember. So how do we avoid these bottlenecks and make the right decision? Again, it's a very hard question to answer, but what we're trying to do is at least answer it in ways that will help the community. Um, there's a phrase here, there's a world of possibilities, and it often evokes optimism and potential, and can universally be seen as a positive concept often. But then some will argue that the abundance of choices can be paralyzing, can lead to indecision, maybe dissatisfaction, and it's the paradox of choice, really. This is no different than at cell and gene therapy. Vectors, you could have an n to the a million or infinite amount of combinations of possibilities of vector design. You're talking about these vectors that have, in excess of 10 kb of vector sequence. All you need to do is change one or two nucleotides and you have a totally different product. So this main message here is that you can have just endless design criteria. So how do you select one? This is the thing. And often people are a bit worried about this and going down this route. So they'll only select one, or as I said, they'll take a kind gift from a collaborator or a stranger and then say, please manufacture this. This is our clinical candidate. That's what happened for years. And as you can see, that sounds, a bit ridiculous in places. But we didn't know any better. 10, 15 years ago, we didn't know any better. We did good science. But now knowing what we know, it's time that we actually take time to really bottom out and choose the vector design a bit more cleverly with rational design principles in place. Good vector practice is something that we're rolling out as part of our own concept. Good vector practice, so it's a bit of a play on GMP or GXP, good vector practice. And we're hoping it becomes a gold standard in the field of cell and gene therapy as well. And the beauty of incorporating a lot of this is that it will capture the majority of these combinations. And it's just a case then of mapping out, say, the budget for it, mapping out the timing, how you actually extend your runway, and build your extend the time up front between that discovery and preclinical animal testing stage. You'll never ever have that opportunity again to go back a step. Obviously, you can, but that's only if you fail. So it's really important that, the main message, take your time at the vector design stage, and it will add value to your overall project overall. Again, yeah, look, it's easy to say that now, knowing what we know for the last, over a decade. But let's be honest, the perfect ATMP developer, and by developer, I mean any scientist, any PI, any lab, any startup company, any developer trying to manufacture a product going into clinical trials. We can't expect everyone, especially startups, for example, to have all of these kind of needs or solutions in place. It doesn't exist. The perfect developer doesn't exist. Therefore, VectorBuilder, someone like that, or a CDMO that will also, again, offer that to you. They'll offer those end-to-end services. Not only will that focus on the other aspects for you, but they'll focus on the aspects that are often overlooked, like the CMC strategy. And that's really important. So yeah, at its fundamental level, vectors are designed to be compliant to the regulation. So it's really, really important then that you encompass all of these needs. So yeah, we'll provide these solutions obviously for you. It brings all the expertise together. Again, these I won't go through all of them individually, but there's some of the main things. And as you can see, I'm regurgitating some of the wording because that's important. It's reminding ourselves that it all comes down to that vector design optimization, not only the vector payload, but the backbone of the virus. Years ago, the backbone was considered not, it didn't impact, it doesn't get packaged inside a virus, so therefore it's not important. But it is very important actually, especially for things like impurities, manufacturing or safety. Unwanted backbone elements finding its way into your final solution of vector once it's fill and finished or sterile filtered, like that's not warranted. So this is now something that we can offer and accommodate everyone in different areas. It's not just literature based, but it's actually based on a few important aspects. I mentioned GVP, good vector practice. So everything we do is based on good vector practice. And also, why can we say VectorBuilder that we can bring this expertise together? Well, numbers don't lie, and the numbers basically that we're in excess of 1 million vectors created now. I think you'd go very far to try and find other companies that can actually have the bandwidth to manufacture this many. And what we do then is we can leverage that in order to instruct, to show better vector design, which elements cause deleterious aspects, which vector elements are, additive. So that's what we're trying to do here and showcase that, not a lot of people would have the same kind of ideas, but can they actually execute it based on what data they've generated already? That's kind of where we hope to stand out. Yeah, so again, this is something that I think is in the spirit of GMP. We have something called the 5 Ps of GMP: product, people, process, and procedures and premises. Again, it's obviously a category. Product is your vector design. People are the people that will manage your project. The process, of course, is the manufacturing process itself, really fundamental to the next slide. Procedures is just how you cannot do anything unless it's controlled and governed by good quality management and documentation oversight. And then premises is obviously have to have somewhere that's GMP compliant to do this work. And that's what we have as well. So segregation, validation, maintenance, obviously making sure that we adhere to GMP practices all the time. And then, yeah, the main take home, that sounds good. That seems like a very much plug and play platform. But unfortunately, every single cell and gene therapy product is different. Even if person X and person Y are targeting the same gene, targeting the same disease, 99% sure that they'll have discrete sequence differences between them, which means that they're fundamentally a different product. That can have, really big implications for manufacturing, as I've said. And the main take home, I think, then is the process equals the product in cell and gene. This is kind of unheard of, where you've got small molecules or monoclonal antibodies or other therapeutic modalities where you have a process, you plug it into a platform, and you can keep reproducing it. It becomes reproducible, robust. But unfortunately, the nature of these gene therapy products and viruses, by their very nature, mean that each process, if the sequence has changed, it means that the product has changed and the process has changed. So, does it mean platform processes don't work? No, of course it doesn't. We utilize platform processes, but a lot of people suggest plugging into a platform process and you're good to go. For people saying that, it's a bit of a misnomer, and it means that you need to really do process development and consider process development a bit more clearly. If it can be modified, as in a certain sequence within the vector, it can cause an effect, regardless of safety, manufacturing, or efficacy. So one way to look at it, again, bringing it back to the kind of gambling analogy, I suppose, gambling in science. Again, it's something that probably doesn't go hand in hand, but it is memorable to the point that you've got a roulette wheel, you're spinning your wheel, as I mentioned. People normally just pick one vector and hope for the best, spin the wheel. Hopefully, I hit the jackpot. What we're trying to say here is by introducing CliniVec early as possible, you're giving yourself GVP options, you're giving yourself more time to select it, better payload and backbone optimization. You're ensuring appropriate steps are made. So basically, you're increasing the amount of vectors that you have. You're increasing your vector runway, meaning that you're effectively putting more roulette balls on the roulette wheel and spinning it. Is it a guarantee of success? No, nothing is in this world, unfortunately. But as you can see, it will increase your go no go chances. It'll extend the time that you have to really do due diligence around that. Obviously that's the benefit, and the whole idea here is that it's not just benefiting, say, you guys or developers or us. It's making people that are investing in these technologies also aware that there is actual tools and strategies out there that will increase the probability of success or reduce the risk in their entire project life cycle, and that's fundamentally it. Um, Again, I don't want to keep harping on about CMC, but this is a very simple schema to show the same as the slide before, but just not in the vector runway. But you can really see as you scale up, you're putting your vector in different bioreactors. It could be single use or standalone cleaning in place systems. Normally, with conventional development, as you scale up, your costs are increasing and your yields go down. Again, I'm in very general here. But with CliniVec development, our whole goal is that you increase the yields and decrease the costs. Every project will be different. In some cases, this will be more pronounced than others. But basically, what we can see is that's the ultimate goal, and I think that sounds attractive to a lot of people. Yeah, and the costs come up time and time again. People will ask, why is there only 28 commercial products? Ironically, 8 of those products are actually probably going to be pulled now, unfortunately, which is sad because they've been cited as commercial reasons. So basically, that means there's no return on investment for the money that needs to be spent to roll them out commercially. Why? Because the cost of this manufacturing or CMC is so high, because they're very unique modalities. They're very difficult to manufacture consistently, so the price goes through the roof. It's very, very hard. So this is a hidden cost sometimes. So if people just do early stage manufacturing with one vector, they can potentially get lucky and it manufactures quite well at a certain bioreactor scale. And then it comes to the phase two and they need more doses, they need more virus yield. And then all of a sudden they go up the next level into a certain bioreactor scale and it just drops off a cliff where the yield is the same, but the cost is actually double. And all of a sudden then you're burning through a lot of campaigns, you're burning through a lot of money. So it's like a hidden cost of unoptimized vectors. Again, more the same story, but just I'm saying telling the story from a different angle. So it's really important that you understand the key metrics. And it's important for business, investors, profitability, whatever you want to call it. It's very influential. So that's the only way these products get made and patients get treated. So we have to manage the expectation of where the money's coming from, and we have to manage the cost. There is a concept we are working on. It's called the price per clinical construct, PPCC. We are trying to give people a bit more of a tool around how they would manage maybe going to an investment committee or a pitching to an investor or pitching to their board, giving them tools that allows them to reveal the hidden cost of unoptimized vectors. Candidate selection, again, this is just the same. I'm talking about the design, manufacturing all in one fell swoop here, but a good way to do it is basically just, okay, I've got one vector. What do? You come talk to us. We'll suggest 15, 14 other vectors. Again, you could have 100 vectors, of course, but trying to be realistic where we would do rational design. We would look at previous data and leverage what we see as a trend. Of course, we have to be very confidential about the 1 million vectors we've made, but we can still see trends. We can still use trends in such a way that minimizes cost and yield deficiencies. So the whole idea, you generate more vectors, and that way you're going to save on the cost of goods, COG, much later. So yeah, look, this is more than just talk. This has actually got real life examples, and we're seeing like the FDA declining to approve certain therapies, citing manufacturing issues. Again, CMC. Manufacturing issues can be quite broad, but 9 times out of 10, it will be linked to the vector design. because that's what you're doing. You're manufacturing a certain sequence. So manufacturing issues goes deeper than just, the facility or the premises, as I mentioned, as one of the P's. Again, look, I won't go through all of them, but it doesn't matter if you're a big pharma divesting. It doesn't matter if you're a small startup. Everyone is coming and meeting these roadblocks, and these roadblocks are a reason why we've come up with CliniVec. We're like, how do we create a tool? What do we have in our arsenal that can help people? And that's where we are right now. And again, look, the FDA, there's the whole developer side of this, but the FDA are actively asking people or CDMOs and developers to have bigger, deeper conversations around vector design, around CMC strategies, and are actually enforcing now something that will help with that, which is this radical transparency of CRLs, complete response letters. This is critical to understanding where these bottlenecks are. But again, we kind of know them already because of our forward facing exposure to the regulations and manufacturing for 10 years. But it'll be really good now because the newsreels will be telling us what we initially thought, which will be vindicating what we've said. Some brief case studies. Again, it's good to see CliniVec in action. So yeah, what we've done here is we've boosted efficacy through promoter optimization. For those who are not vectorologists, I suppose, or molecularly inclined, promoter is what drives the gene of interest. So the promoter is fundamental to the success or the expression of that gene in situ. What we've done here now is we modified the vector in certain ways that enhances the overall therapeutic efficacy. So this, it says a lot here, but ultimately what that means is we've got a client that came to us with one vector that could have been a kind gift that they don't know what to do with next. Do I change the promoter? Do I think it's okay? Will it work at this dose? Is it safe? Does it create kind of integration events? And fundamentally, does it impact the host cell, hex cell, which is the host cell that you manufacture in? Because if that doesn't has a decoherent effect with the host cell, well, that's where your manufacturing and sequence issue comes from. So that's what we try and do. That's what we try and understand. Is the promoter going to be efficacious as well as can we manufacture it? And what we've done here then is we've enhanced overall therapeutic efficacy, and we've demonstrated that it's easier to manufacture as well. Second case study. Again, sorry, I touched on efficacy, but this is on the manufacturability side where we've got lentivirus. Again, agnostic to which vector design, can be AAV, lenti, adeno, HSV. We manufacture all of these, of course, but this is a lentivirus setting where we've gone from having quite low transducing units in a harvest. And what we've done then is we've modified and added prime elements during CliniVec consultation and design modifications. And ultimately what that led to is, almost a log fold increase in functional LV titer at harvest. And this is obviously especially good for companies that are in the ex vivo or in vivo CAR T space, and where you get a higher rate of positive modified T cells. So again, there's a huge difference between 16 and 41%. Again, it's all about the efficacy, getting more bang for your buck, and yeah, getting more out of it. And that's what this is demonstrating here now in two obvious case studies. So yeah, we're just closing a little bit now. That's CliniVec. Again, why we're able to say a lot of this, we are a global leader in gene therapy technologies. We have a comprehensive vector hub, which is almost like a marketplace. You just go in, you can place your orders. You don't have any interaction with any sales team if you want. And we pride ourselves on the fact that if you know what you're doing, we have given you the marketplace to do so. And if you don't, by all means, reach out to us and we can be accessible and help you in creating these vectors. We have everything from research to clinical grade, as I mentioned. And then some key numbers, 1.2 million vectors designed, literally 400,000 vector components, individual components. So again, could you use all 400,000 of those in a vector? No, there's only so much space. But at the same time, that gives you the optionality, and that could be quite daunting. We have many partners. We partner with academic institutes or industry partners, about 4,500, which is great and growing every day. And then, yeah, a lot of our materials have been used in publications, including Science, Nature, and Cell. So it's a real testament to the materials that we manufacture as being good quality. And yeah, we're proud that we can facilitate the community and continue to do so. Um, yeah, this is more of a slide as a pitch slide as where you show all our four major segments. Again, research grade solutions, manufacturing to GMP grade solutions, CRO. Again, we do more than just manufacturing. We work with animals, of course, if you need to. We need to actually generate libraries, capsid engineering, you name it. We pretty much do everything. And what we're trying to do now is we're looking at maybe out licensing some of these wonderful assets that we have. So if anyone is looking for novel capsids, novel promoters to make your product kind of better straight out of the packet, and by all means reach out to us in that regard as well. Yeah, and then summary, CliniVec, yeah. So it's the whole idea is that we're here to enhance your success. It can be looked at in a few different ways. CliniVec can mean something slightly different to everybody. It's it's a free consultation service. Again, people love getting yeah, free consultation, and that's the thing, you can always get advice from somebody, and I think that's what the whole field needs. Maybe if we worked a bit more collaboratively than in silos over the last 10 years, we would have actually not seen as many failures or issues in the valley of death. So again, it doesn't matter, whatever's happened in the past has happened, we can learn from it, but it's all about going forward. um Things like payload configuration, tissue specificity, reducing toxicity, enhancing manufacturing. I don't want to harp on about it, but ultimately reducing the cost of goods. These are the hallmarks that will make the better developers over the next few years, and we want to help them get there. So yeah, that's pretty much it. Again, by all means, reach out to me on any of this. And yeah, pretty much thank you for your attention. We have some other kind of key educational materials as well that kind of showcase the different aspects that we have. By all means, you can find those links at the conclusion of the webinar. And yeah, that's it for today. So thank you very much for your time, and hopefully, yeah, hopefully you learned something today, and that we're here to kind of help and facilitate in any way possible.
Thank you so much, Alan. That was very, very comprehensive. If anybody has any questions, please feel free to type in your questions in the little Q&A box, and I will be able to read them out, and hopefully you'll get your answer. We have a couple already, so I'm going to go ahead and start the question and answer session. The first question we have is, what elements or aspects of a vector are most important?
There's, as I mentioned, there's 400,000 elements. You could probably ask 1,000 different people and they give you a different answer. So unfortunately, Sunita, it is quite subjective. Um, the main, I'd actually look at it from the other side of things, where what's the most important. It's not necessarily what in discrete element is most important. The main thing is the principle of why you're designing something. Do you have extra elements in there? We see a lot of clients having extra bits and pieces, kind of extraneous bits of DNA that might be legacy, carry over. And they're like, well, it doesn't make any difference. It doesn't harm my product. And it's like, well, the FDA or MHRA or any other EMA will look not favorably on that, because they will say, well, if it doesn't serve a purpose, why is it in your final vector design? So I'd actually answer that question on the reverse, where we take principles to your vector design. If it doesn't belong, eradicate. If it does belong, make sure you create multiple versions of the same element you've selected and lock each element as you go along. So if you're choosing a promoter, a poly a gene of interest, look at different promoters, look at codon optimized versus wild type, look at different poly A's, because they all make the configuration, and each we've shown that different configurations, even small changes, can make huge differences to the efficacy in manufacturing. So yeah, that's the best way I'd answer that. So keep an eye out for if it's extra and if it's not meant to be there, rationally remove it rather than trying to focus too much on what you should include. Yeah.
Thank you. Our next question is, does CliniVec service include the support at research grade stage?
Yeah. Great question, because that's the thing. A lot of people will see this and hear manufacturing, they'll hear GMP, and be kind of put off. Or if you're not in that world, you could be quite, it's quite a mystifying space, GMP manufacturing for clinical use. Geez, that means serious business. So what does that mean? But the whole idea of CliniVec is to act as a bridge between research and clinical trials. Why? It helps the, obviously, hopefully it helps the developer, but let's be honest, it helps someone like the CDMO too, or vector builder, because we can de-risk you handing us a vector if we have already got eyes and ears on that vector and we've been part of that journey. Plus, there's the continuity factor. A lot of people now are like, well, I've done a lot of research over here. Now I need to go talk to a CDMO that I've never really talked to them. Like, they talk a different language to me. They're talking manufacturing. Whereas by the time, a year or two into the research project, we'll be talking about CliniVec, we'll be talking about CMC. You'll already be kind of a partial expert yourself in that field. So then it becomes demystified. And that's the whole idea there, that reach out to us, even at the research stage, and we can help. Why? Because we hope maybe you're successful in animal testing and POC, that you come knocking on our door a year or two later and you're like, I might as well stay with you guys now. You're manufacturing, you know what you're doing. Please continue to use, I want to continue to use you. So that's the answer to that question, Samita, is, by all means, yes, you can reach out at any stage, anybody. The door's open to everybody at every stage of the development life cycle.
That's wonderful. We have another question. Any recommendation for the discovery phase? Does VectorBuilder have vectors available so that we don't have to depend on strangers?
Yes, we do. We have off-the-shelf vectors that are ready to go. Of course, we have a range of, IP based ones, or we've non-IP based ones, depending on the budget. We have a lot of big pharma clients, of course, so we're trying to cater to people in different ways. You don't have to use a proprietary backbone. We, of course, have them because we know they perform a little bit better. But back to what I said, process equals product. That it just because you use a kind of set backbone is not a guarantee to success. So during discovery, let's kind of answer those questions together. It's never too soon to try and figure that out. But yes, we have a whole host of, we can design them. We have a very short turnaround time for vector cloning. That's how we made a name for ourselves. We build vectors. But also, it's about understanding the client's needs as early as possible and working with them in such a way that, yeah, do you need an off the shelf? Do you want to create something from scratch? Do you yourself want to create some novel IP? We can help with all of that, and we've done so with our thousands of clients.
Thank you. The next question is very related. You probably answered like parts of it, but there might be some parts missing. What are the outputs of CliniVec? Will you provide us with novel sequences that we can test? How does this process work?
Yeah, you can provide you with novel sequences. We provide you with FASTA files, with sequencing files. We can give you schematic maps. Of course, we use Vector B, which is obviously a very analogous product to Snapgene. A lot of people are users in the audience might use Snapgene, but we obviously use Vector B, which is a free version of Snapgene as well. So these are all the things how these files are created, and then these become shareable files with all in any way you want. We can package it in a few different ways. Or in some cases, some of our CliniVec clients, they've requested that myself or some of the team actually attend some of their meetings or some of their senior meetings to explain or demystify the vectorology or the manufacturing of what we've made for them. Again, when you think about it, it seems like a simple solution. Why don't, instead of feeding off the information that we give you, we're seeing now more and more people. Why don't you just join the meeting yourself, and help explain? And that builds the relationship. It allows us to, showcase what we know and what we can do. But the answer is, yeah, we can do multiple formats in any format that's needed. Yeah.
Thank you. So this person is still doing in vitro research, but the results seem promising. They're wondering if it is too early for CliniVec.
No, never. So answered that question a few times there now, probably just in different kind of contexts or syntax. But yeah, anytime, now is the time, because you're getting to a point where you're locking in on your vector. Point of no return is fast approaching. Now is the time to have the conversation. And it doesn't mean you have to scare you and we have to change it. In some cases, it's just a validation tool. The guys at VectorBuilder have looked at it. They've assessed the vector for me for free. Now we know that we're not missing anything, or it is actually designed really well. Like, that's happened a good few times as well. So think of it as a safety net or a sanity check of it. And If it's too early, we'll know after the first 10 minutes. And then what we can do is we can follow back up with you in a few months' time when you have some new data or you have something new to share. Or we can be part of that entire journey as well.
Wonderful. What are the most common vector design issues related to clinical design you repeatedly see?
Yeah, a lot of people have those superfluous regions or extraneous bits of pieces that are carried over from maybe 20 years working in a lab, or again, that kind gift mentality where I know someone that has a good vector, I'm going to send it to them. And then you do sequencing or you understand the map and you've got like F1 RE regions in there. You've got the classic cases. You've got ampicillin backbone where, ampicillin is pretty much, it's not allowable that you go through clinical trials with ampicillin. Why? Because it's obviously a beta-lactam related antibiotic, which means that people out in the public or people out in the global populace can have hyper beta-lactam hypersensitivity. And they could say, yeah, well, it's a few degrees away from the antibiotic. We're purifying it. We're making virus. It is a few steps away. However, that trace elements, and it's hypersensitivity for a reason, that you can have trace elements of your ampicillin coming through into your final product. And the FDA will turn around and say, well, you're meant to change it. Why not just change it instead of trying to justify it? So that's what I see commonly where we have CliniVec clients and they're like ready to go. And we've done a bit of optimization at the research side. And then they're like, so do I need to do the kanamycin switch? You're like, yeah, you do. And then what happens is we're now catching people earlier where they're doing the research work with the kanamycin backbones. So that means then there's literally that continuity factor. It tells a nicer story that you've used the exact same sequence at the discovery or research stage than you have at the later stage. And that's been one of the challenges of CliniVec because it's the timing. And that's why the answer to that person's question is earlier the better because you could be using ampicillin derived backbones, and that's fine. Obviously, for screening, for other applications, that's fine. We're not saying they're bad, you should never use them. But if you have any inkling about going to clinical trials or preclinical work, um, you have to revise the ampicillin. So ampicillin would be probably the biggest issue or vector inclusion that we see, Samita. Yeah.
Great, thank you so much. We have one more question. You listed many aspects of AAV vector optimization. What are the ones that you think are most important to consider first?
Mm, God, yeah. A few different ways to answer this. It is context dependent. It really understand your mechanism of action and understand your pathology. Go beyond the literature. A lot of the literature, it's great and the data is quite good, but you can almost tell that the literature has been based off a vector design that worked. But is there any optimization, such as various different iterations or combinations of vector gone into that? Probably not. So, the answer is very, very broad. Like for me personally, I've seen huge differences when it comes to the promoter. I've seen huge differences when it comes to making sure that the ITRs are kind of in a more stable form. Again, we're very excited about a new product we have coming soon, which is, a kind of ITR stabilizing asset that will pretty much allow any AAV vector to not be truncated, not have their ITRs truncated, because the ITRs are obviously an intrinsic palindromic region that constantly gets silenced or truncated or deleted in bacteria cells when you're fermenting or manufacturing them. So obviously that's an intrinsic issue that it's very hard to manage. Whereas now we're trying to maybe install an AAV insulator type backbone that will avoid those pitfalls in the future. So yeah, they're the things that I've seen make the most different. Again, there's been situations where codon optimization has worked really well, but then likewise, there's been a lot of situations where codon optimization has been quite negative due to maybe integration issues or safety issues later. So yeah, it's a very hard question to answer, but the best way would be to say that every element inside is important for the same kind of good vector practices that I mentioned before. Yeah.
All right, thank you. We have one last question. What is the most important thing for regulatory agencies from an IND submission point of view?
Yeah, having a story, and again, I don't mean storytelling, I mean having the narrative or the provenance of everything that you've done very clearly understood. You used to have startup companies and they'd have predominantly scientists and they'd go and present the data. It's very amazing. It's revolutionary in some cases. Um, and using the vector example again, they'll say, so your vector works really well. Congratulations. But what's with all this extra junk inside of it? And I was like, well, yeah, we were afraid to go back a step, so we didn't really know what to do. That lack of decision making or that lack of clarity, that's what the regulators are fearful of. That we're smart people in a smart sector making smart products, smart therapies, but we show really poor decision making attributes sometimes. And I think that's what they're the most important thing. Of course, I can sit here and say your module three, your manufacturing, your animal study, statistical significance, safe, all of that stuff is very important. But fundamentally, they won't be able to check absolutely everything. So it's really important then that they get the feeling from you that, hey, these guys, they're good decision makers. They know what they're doing. I'm more and more confident that they've done all the right things. They've crossed the T's and dotted the I's in every way. So for me, that's my own personal experience. And you can have those conversations with the regulators. They're not there as a police. They're there to be engaged with, which is another failure, I think, within the field that people, I don't want to mention this just in case they say something wrong or they ask me to do something I don't want to do. That's a recipe for disaster as well. You have to engage with them. Of course, if you're worried about that, use CliniVec as the initial tiered approach to that. Use us as the initial sounding board to have that conversation. That's what it's there for.
Yeah, as an extension to that previous question, from a business point of view, what do investors want to see?
That as well, good decision making. And yeah, they want to see that the candidate selected portrays, that you've selected correctly, that you've taken your time selecting. It's a bit counterintuitive because I know, having had the pressure or working under duress from certain investors, that they want the candidate selected to meet a certain milestone ASAP, as in we wanted tomorrow, or yesterday, in fact. Um, but I hope now that the failures in the field, the clinical holes, the learnings that we collectively have as a community makes them realize that actually you shouldn't be asking for them to have candidate selection ASAP. It should be critical, compliant, vector decided through iterative deduction. So that way it's actually selected based off, you're not putting pressure on them. That way you're not having them work under duress, because a lot of people are obviously afraid to go to investors and say we're delayed, we're delayed. So really go and set out a clear path to vector and candidate selection. And that's the whole basis of vector CliniVec as well, and vectorology.
Great, thank you. We have one more question come in, if you're up for it.
Yeah,
How
yeah.
does using kanamycin selection or other antibiotics compared to non-antibiotic selection systems in antibiotic-free vectors in terms of manufacturability and DNA production yields?
Yeah, from everything that I can see in the field right now, I see equivalency. Do I think they're better in every case? No, they're about the same, which is, I mean, that's acceptable because you're removing one safety concern and getting the same performance. For me, that's a win. But yeah, the whole idea now, people are now wanting to. Like, kanamycin's obviously a good example where you switch from ampicillin to kanamycin, but the FDA also state quite clearly in the annexes that you should consider antibiotic-free selection or vectors as well, because then that way the risk is reduced to zero. There's not many systems in place like that. We have a few ourselves, MiniVec, of course, but there's nanoplasmid, there's a few different other versions out there, but the doggy bones of this world, these are all useful modalities that will kind of help with that. But having that clinical acceptance is something that's yet to happen in that kind of field. But it's happening. The next 2 or 3 months or years will show that these vectors are very useful. But regarding performance differences, they're more or less equivocal, and that's okay. It's a very general answer, I know, but it is the way I see it and the way I see the trend.
Thank you. That was the end of all the questions that we had. And thank you so much, Alan, once again. That was an incredibly informative presentation and a webinar. Thank you so much for speaking about CliniVec with us today.
No worries. Anytime. Thanks
Awesome.
for everyone's attention.
All right, everybody. We will hopefully see you in our next gene delivery seminar series. Bye-bye. Good night. Good morning, wherever you are. See you soon. Bye.